Hi biologists, let's start by looking at the learning objectives for this lesson. Take note that this material is for the higher level biology only. At the end of this section, and following the biology syllabus, you should be able to use the active site theory to explain specificity. What does this actually mean? What are we trying to understand? Well, it's straightforward enough. We have to use the active site theory to explain specificity. In other words, why enzymes will only react upon one substrate or substance. Let's sort higher level enzymes by first looking at the active site theory. The substrate is the substance upon which an enzyme reacts. SUB in substrate, SUB in substance, represented here in the pink. The product is the substance the enzyme forms. In other words, the substance that's produced. Again, product and produced. The active site is that part of the enzyme that joins or matches the substrate, seen here. You might notice, to keep life easy, I have made the shape of my active site like a letter E on its back. Now, the original active site theory stated that the active site had a rigid shape, an inflexible shape. It stated that the enzyme had a rigid shape also. The enzymes functioned according to a lock and key model. The substrate fits into the active site like a key fits into a lock, and they had to be an exact match for the reaction to be carried out. However, it was discovered that the active site is a flexible depression on the enzyme surface. In other words, it's a hollow on the surface of an enzyme, and there's a certain flexibility attached to it. It is not a rigid shape. It's actually created because the enzyme is a globular protein with a three-dimensional or a 3D shape. And this 3D folding of the globular protein gives a three-dimensional hollow, which is rather flexible on the surface of the enzyme. It was also discovered that most enzymes are made of more than one globular protein that are called domains. And in actual fact, these variable domains, the globular proteins, can vary, so they join together to form the enzyme overall structure. Now we will use the active site theory to explain enzyme specificity. This is a very important idea, and they are very fond of it in exams. It is a learning objective of this lesson, so you have to make it your business to understand the idea of specificity and learn the meaning of the term. Specificity refers to an enzyme's ability to react with only one substrate. It is specific to a substrate. It will only react with one particular substance. Therefore, you could say enzyme activity is specific for one reaction. Why is this the case, and what exactly does this mean? We have to explain this in terms of the active site theory. Enzyme specificity. Enzymes only react on one substance due to the shape of the active site. As we've said before, enzymes are globular proteins. They have a three-dimensional bulky shape. The 3D bulky shape of a protein means that the active site shape will be three-dimensional as well. The 3D active site shape means that the enzyme will fit and only react with a substance that has a complementary or matching active site shape. And this is the key sentence that we have to take on board. And you can see that what we're doing here is that we're using the idea of the active site to explain why the enzyme will only react on one substrate. And there you have it. Now that we've reached the end of our lesson, have we achieved our objective? Can you use the active site theory to explain specificity?